Alrighty, hello. Welcome everybody. It's promptly 2 p.m. where I'm at. It's week 4, and this week we're going to be discussing the immune system. So go ahead, grab out your worksheets, and we're going to go ahead and get started. Alrighty, so first we're going to look at how the immune system does what it does, and to start off, we're going to be just looking at some terms and going through and just saying their definitions. Hello and hello to the two people in the chat. So, first term is bacterium or virus particle. The definition of this is simply just types of pathogens. The next is helper T cell and a receptor helper T cell. And this is a type of immune cell that stimulates killer T cells macrophages, and B cells for those immune responses. Cytotoxic T cell is a T lymphocyte, which is just a type of white blood cell that kills cancer cells which are cells that are infected by intracellular pathogens or cells that so it kills cancer cells, cells that are infected. The way that I wrote the definition earlier made it sound like that was the definition of cancer cells, so sorry about that. So T lymphocyte, a type of white blood cell, kills cancer cells, cells that are infected by pathogens, or other damaged cells. A B cell is also a type of lymphocyte that are responsible for humoral immunity. And that's just a part of the adaptive immune system. And so these types of white blood cells produce antibodies which are a huge part of the adaptive immune system. Memory T cells are what are called antigen experienced cells, which means that they've encountered this antigen before, they know what to do when it comes again. And so these initiate faster and more potent responses when repeating an encounter with that antigen. Okay, a memory B cell is a type of B lymphocyte, also part of the adaptive immune system. They develop within germinal centers of the secondary lymphoid organs. And they circulate in the bloodstream in more of like a quiet state, just kind of monitoring, looking around, and then sometimes they can do this for even decades in our system, which is really cool. Cytokines, they have, their main role is just to have an effect on other cells. So any number of substances like an interferon, interleukin, growth factors, anything that's secreted by certain cells of the immune system are cytokines. So they have an effect on other cells. So I'm just going to put interferons. And then discussing more on those interleukins down here, they are a type of cytokine, and they were first thought to be expressed by leukocytes alone, but have also been found to be produced by other body cells. So I'm just going to add produced by leukocytes and other body cells. And that's all you really need to know for this guy. Alright, CD4 protein is a glycoprotein that serves as a co-receptor for the T cell receptor. So what this is it's found on the surface of immune cells like T helper cells, monocytes, macrophages, dendritic cells. It just helps for being a receptor of different kinds of pathogens that could infect our bodies. All right. Moving down. So MHC1 and 2 molecules, to explain this, I'm going to break it up into either 1 or 2. So class 1 MHC molecules, they span the membrane in almost every cell in an organism. While 2 are restricted to cells of the immune system called macrophages and lymphocytes, so found only in macrophages and lymphocytes. Alright, antibodies. I'm sure you guys have heard these before, but it's a blood protein produced in a response to a specific antigen. And what they do, just to give you a little bit more of an in-depth definition, it's not on the sheet, is that antibodies combine chemically with substances, and those substances which the body recognizes as foreign, not from our bodies, things that we haven't seen before, things potentially dangerous such as bacteria, viruses, other foreign substances in the blood. So I'm just going to say that recognizes foreign and potentially harmful substances. Because I can't fit substances, I'm going to put things, which is a more simplified word. Alright, an antigen is a toxin or other foreign substance that induces an immune response. And we know because antigen, we're especially just talking about that production of antibodies. An epitope is the part of an antigen molecule, so these guys are kind of related. It's part of an antigen to which an antibody attaches itself. So it's part of the antigen, but it's where the antibody attaches. An antigen and antibody, though they are very similar and very different, it definitely is hard for me sometimes on tests to immediately, like, you think like, okay, yeah, I know what an antigen is, I know what an antibody is, but when you're just given a test, that kind of pressure definitely for me can make me lose those kind of simplistic things. So just try to remember antibody as protecting your body. I like to put like antibody, my body. It's protecting my body from antigens. Or, yeah, that's it. No or. That's all I had. All right. The thymus is a primary lymphoid organ of the immune system. And so what the thymus does is that within it, thymus cell lymphocytes or T cells mature. So primary lymphoid organ where T cells mature. And as we've said before, T cells are super important to the adaptive immune system. So that's where the body adapts specifically to foreign invaders. We've seen them before, and once we see them again, they're toast. All right, bone marrow. Bone marrow is that little spongy tissue that's inside some of our bones. It's in, for example, it's in our thigh bones and our hip bones. That has a lot more than other smaller bones like carpals in your wrist don't have as much. But it contains, so the bone marrow contains stem cells. And stem cells can develop into red blood cells, and we know red blood cells carry oxygen through the body, or can develop into white blood cells that fight infections. And finally, it can also develop into platelets, which help with blood clotting. The hypothalamus, also a huge one. I think maybe some of you guys have heard before. It's in our forebrain. It's the region of the forebrain that's actually right below the thalamus. And it coordinates the autonomic nervous system and the activity of the pituitary. And so what this does is it controls thirst, hunger, other homeostatic systems. So I'm just going to write controls homeostatic systems. We talked about how the hypothalamus, or the medulla, controls our respiration rate, so we can think about this in terms of sleep, emotional activity, hunger, thirst. Things to maintain homeostasis within our bodies is what the hypothalamus does. All right, so we just have a few more definitions on the next page. I'm going to scroll over. A fever is a temporary increase in body temperature, which is often due to illness. Having a fever is a sign that something's out of the ordinary, something's going on within your body. Clonal expansion is when an can what? Be cured with more cowbell. Sorry, mine's a little more delayed than your guys's is, so first off, I love the reference. I'm just sad I didn't know what it was to. I own the DVD with that whole Will Ferrell SNL best of, and it's incredible, by the way. Best DVD I've ever bought. The Fever. Oh, yes, sorry. Okay, I was like, the hypothalamus? What? Anyway. So clonal expansion is when an antigen drives the that it's spelling. The proliferation and differentiation of cells with specific antigen receptors. And then finally, self versus non-self is just the key difference between self and non-self antigens, where sorry, I can't talk right. Or self and non-self antigens, where cells of our own body are known as self antigens, and then the antigens that aren't original to our own body are called non-self antigens. But to remind you, an antigen is any substance that induces an immune system to produce antibodies. So antigens can come from the outside or they can come from within ourselves. Whew, okay, a lot of definitions. Since it is 2:20, I'm going to go ahead and put in that form within the chat. I'll put it in a couple of times from this, just because I know some people can miss it. So, but yeah, it's just there if you want to fill it out, and I'm going to move on to question 1. So, what are pathogens, and why do we need to prevent them from colonizing our bodies? If pathogens do manage to colonize, what effects can they have? So, pathogen is just a fancy term for any disease-causing organism. Pathogens can include bacteria, viruses, fungi, and protists. Bacteria, viruses, fungi. So the cells of our bodies contain every component that is required for life, and these components support the lives of our own cells, but kind of as a trade-off can also support the lives of pathogens. So pathogens can compete with our own cells and potentially destroy them, and that answers that. So we've answered what are pathogens and why do we need to prevent them from colonizing our own bodies is because they can take over our machinery and kill our own cells, and we don't want that. I'm just going to put bad and then in small things for us, because it's obviously good for the pathogen. And then, to answer the last one, if pathogens do manage to colonize, what effects can they have? So they can overgrow, putting pressure on our membranes and cavities. I think that parasites do count as pathogen. Sorry, I just had to contemplate. So right. So though the parasite is an organism that lives on or in another organism or species, parasites can often form a type of symbiotic relationship where they're not necessarily killing their host but using their host as a way to survive. And so I think that parasites can sometimes count as pathogens if they are disease causing, but they're also not exclusively known as pathogens. What we mainly consider as pathogens are things like bacteria, fungi, protists, and viruses that cause disease. But a parasite that causes disease, it's kind of like the square, rectangle, rectangle, square. So a parasite can be a pathogen, but a pathogen is not always a parasite. Does that make sense? And the student in the chat, thank you so much. That is also a great explanation. Okay, awesome. Yes, of course. And thank you, students, always for helping each other out in the chat. That's always really great to see. So they overgrow. They can put pressure on our membranes and cavities. And an example of this is if there's a disease in the ear, like an ear infection, eardrums can rupture from this kind of pressure. And another example is within the sinuses. So the pressure builds up in the sinuses, and that can cause blockage and pain. So if you've ever had a sinus infection, which, I've had sinus infections, and they are just the worst when they're bad. But I could hear almost cracking by my nose where the pressure was building up so much that it was trying to relieve. That's another story. Sorry. But yeah, so you can build up pressure also in your sinuses. Effects, they can produce toxins that destroy our cells. I'll put these in parentheses because they're examples. And some examples of that are just like bacteria that produce exotoxins that can destroy our cellular structure. One of the last effects I'm going to talk about, there are definitely more effects that pathogens have when they colonize, but just the last one I'm going to say that they can destroy cells' normal function. And this is like many fungal infections. One example is athlete's foot. And this is like your question earlier in the chat. So athlete's foot is actually parasitic. So they can parasitize cells and destroy their normal structure. And funguses that parasitize are really, really good at being pathogens. Fungi is awesome and beautiful, but also kind of really scary. So, yeah. Moving on to number 2. What general defense mechanisms does the body use to help prevent colonization by pathogens? For example, what general defense mechanisms are involved in local inflammatory responses? Some general defense mechanisms to answer this first question up here would be skin and mucous membranes. There's a lot of secretions on the skin, for example, that act as physical barriers to the entry of pathogens. So lysozymes that are on our skin can provide an acidic pH, which inhibits the growth of a lot of microbes on the skin. That's why maybe when you've had a lovely night of crying, your face actually ends up feeling really, really soft. If you walked out of a sad movie and you were drying your tears out, and you're like, wow, my skin feels really good. It's because there's lysozymes in your tears, and they might be breaking up bacteria on your face. So let let that river run if you're ever crying. You're giving your cells on your skin a nice cleanse. But yeah, these are physical barriers. And I'll put lysozymes from tears as an example. And then to answer that second question, for the example of the inflammatory response, at a localized injury, wherever the cut happened or there was, a sliver or something, the blood is exposed, you need an inflammatory response. You want to cut that blood flow out from going where it's not supposed to go. So at a localized injury, things called prostaglandins and histamines are released. Not to get histamine confused with histidine, which is an amino acid. Prostaglandins and histamines cause increased blood flow and swelling to the area. And I know this sounds kind of intuitive, but this all has to go with that clotting mechanism to prevent invasion by additional pathogens. If we're flooding the area and not just like taking everything from the outside in, it prevents extra things from coming inside. So this causes increased blood flow and swelling. Nope. Swelling has two L's. To prevent pathogens from entering. Additional pathogens are not allowed to come in through that, and then what happens is phagocytic white blood cells will move into that region really quickly and then consume whatever pathogens are there to prevent colonization. So now that we have this little river that we've created with our swelling and the blood, white blood cells move in, and those phagocytes consume any pathogens to prevent them from colonizing. And if you've seen pus at any sites of infection, I remember getting into accidents a bunch when I was a kid just from riding my bike or running outside or climbing trees or whatever. And if it was a big gash, it was treated over a while, we would see pus and kind of gross stuff, but that's actually just an accumulation of those phagocytic cells, which is really cool. There's also a note in here I got from professors that there's, in chapter 43, there's a more detailed description in your textbook of this kind of inflammatory response for a cut or a wound. I've always found this really cool. You have a bunch of fighters in our bodies that are there to protect us. No, actually, pus is not bad. It's a good sign. It's a good sign. What that means is that pus, that little white pus, is just our white blood cells that are going there to engulf any bad things from going into our body. So pus is actually a good sign. I mean, it looks gross, but it's really good. It's doing a great job. It's kind of like mucus, how that's catching all the bacteria. We're swallowing it, so it goes into our stomach and it can be digested. It's just another immune response for us. So it's a good thing. Yeah. I really like this stuff. I like the immune system. This is kind of my ish. So I'm happy that I get to teach it to you guys, or help you guys learn a little bit, at least. So yeah, okay, cool. But it needs to be cleaned. Yeah, so it's good, obviously, if the pus is too much, to clean the infected area. You're not doing anything bad by cleaning it away at all. Running over water and everything is another mechanism we use to wash that kind of stuff away. But yeah, That's why maybe if infections aren't cleaned so often, there might be even more pus there. Now that I'm saying that word so much, it's becoming like kind of gross to me. But yeah, exactly. So now we're going to go to looking at the differences between B cells and T cells. I'm going to start with, yeah, of course, of course. No need to think. Always ask your questions. I'm going to start with the B cells, and then I'm going to go on to T cell responses. So we're going to start on the left-hand side. Um, Individual B cells respond to different types of foreign antigens by secreting antibodies that interact with those antigens to cause them to clump together. So that's the first point I'm going to write. So what this does is the clumping, or what's called agglutination, or to agglutinate. Um, so agglutination basically tags or identifies the antigen for removal by phagocytes or macrophages. Other antibodies can interfere with the function of antigens by binding to them and blocking their action. So this is another way that B cells can do this. So they can actually, antibodies bind to the antigen itself to stop it from functioning. So stopping its function is just stopping all of its actions from further continuing within the body. Okay, T cell responses. When a body becomes infected with a pathogen, a piece of foreign protein from the pathogen interacts with an MHC molecule. We talked about MHC molecules just to give you a rundown. There's MHC 1 and 2 molecules. Class 1 span the membrane of almost every cell in an organism, and class 2 are restricted to the immune system cells of macrophages and lymphocytes. So that's just what an MHC cell is. I'm just going to write that down. And what this does is that the MHC antigen complex, so that's what we call it when that foreign protein is bound. So a piece of foreign protein interacts with that MHC molecule. We know that those are receptors on cells, so that is what now becomes the MHC antigen complex. That MHC antigen complex migrates to the surface of the cell's membrane, and individual T cells that have the complementary antigen receptors bind with what is displayed on that MHC. And then to be specific, T cells that have a complementary antigen receptors will specifically bind to the MHC2 complex, because the MHC2 is what is found in the immune system. But cytotoxic T cells bind to the foreign proteins on class 1 MHC molecules. And then as a note, what cytotoxic T cells do is kill the infected cell. T cells respond T cells respond by dividing and producing activated helper T cells and memory T cells. The helper T cells produce cytokines, which help the B cells to differentiate into antibody-producing cells, and they also help cytotoxic T cells to become active and killing the infected cells. So memory T cells are just to remember the antibody further. Helper T cells produce cytokines, which then promote B cells. And they also produce cytotoxic T cells. or they activate cytotoxic T cells. Okay. I know T cell responses. This was a huge crash course on T cell responses. My advice for this is, instead of seeing it written out in the way that we do here, is to just look up a diagram, because I feel that going through, seeing the pathogen latch on to a receptor, seeing how that receptor is either class 1 or class 2, how class 1 recruits cytotoxins, just kill the cell. 2 then recruits T cells, which get helpers. Helpers recruit B cells for further adaptive immune responses. They recruit cytotoxic T cells to help with killing, and they produce memory cells. So it's kind of a branch into a branch into a branch. Okay. About 14 minutes left, 2 questions to go. We got this. I'm going to put the form in the chat just one more time. So, if about 10 to the 5 genes are available in the human genome to produce proteins, how can we produce 10 times 10 to the 6 different kinds of AB receptors on B cells? So helper T cells. Sorry, just a question in the chat. So cytokines, remember, are a bunch of different cells, like interferons, interleukins, growth factors, and those are secreted by certain cells of the immune system, have effects on other cells. So what the helper T cells produce, you're right, just to go through it, the helper T cells produce cytokines, and those help appropriate B cells. So they don't, yes, recruit. So what those do is recruit B cells to differentiate into antibody-producing cells. So the T cell recognizes the MHC2 protein complex with the antigen, and it's like, I need help. I need somebody to remember. I need this. So first it does helper T cells recruit. I am making this. I'm going to start over once more. I'm so sorry. My mind is like going a million places, but I have it. I have the spirit of it. So you're right. Helper T cells produce cytokines. Those cytokines recruit B cells. Those B cells help to differentiate into antibody-producing cells. So that's the end of that line. Cytokines get B cells, come over to help, and the B cells are like, okay, I'm writing this down. We're going to get antibody-producing cells. We're going to get a bunch of antibodies out here so that if we see this antigen again, it won't happen like that. What the helper T cells also do is activate cytotoxic T cells. Those cytotoxic T cells are what actually kill the invaded cell. The helper B cells are just to get the information, to be like, okay, I'm getting the name, the address, the face, social security number of this antigen, just so if we see this person again, we can book them. But then the cytotoxic T cells, like, I just want them off the planet, that kind of thing. So I hope that helps. Does that clarify? Yes, the cytotoxic T cells. So, sorry, the cytotoxic T cells are already there. The only thing that the helper T cells produce are those cytokines. I know that that's the cytokines, and cytotoxic is hard, but the helper T cells produce cytokines. They also recruit cytotoxic T cells. Really quick, I have an empty page here. Makes cytokines. So does this help, this little diagram? Okay, awesome. Yeah, I know the way that I wrote it out was confusing, so thank you for having me break this down, because I think that this makes it helpful for everybody, including myself. I was like talking in circles. So, yes, awesome. I'm really happy that this helps. And I will definitely try to get this posted as well. Alright, so about those genes, how can we make so many different kinds of receptors or proteins on B cells, but only have a certain amount of genes? And how this happens is that each of the variable regions of the antibody on both light and heavy chains, so you know how an antibody kind of looks like this, and we have the heavy chain, the light chains. It's not cute, but anyway. So each of that is made up of 3 parts. So variable region, each is coded by a different region on the DNA. The DNA segments for the 3 separate parts of each variable region can undergo random recombination in the production of B cells. And so as a result, fewer genes are required to code for that huge number of different kind of receptors, because we have 3 different places that are variable, and on top of those 3 different places in 3 different sites on the DNA, those different DNAs can recombine. So there's like an infinite number of possibilities. I'm also hearing this diagram is great, awesome. When in doubt, I'm gonna make a diagram. I don't know why talking is always my first solution, because it just generally confuses me and you guys. So I will definitely try to make more diagrams moving forward. Yeah. Cool. And then the final question, how does HIV affect the immune system? So human immunodeficiency virus targets cells that display both the CD4 receptor and the chemokine receptor. So I'll say targets cells. And the thing about CD4 and chemokine is that both of these receptors are found on helper T cells. And what this does is that it ultimately leads to the death of helper T cells. And so that really destroys a lot of the immune responses that are triggered. And as we saw, the helper T cells really do a lot. They get the guys that are going to kill in there, they get the ones that are going to remember in there, they help recruit a bunch of cells. Without them, we don't even really have the memory B cells coming in. And so when HIV affects these helper T cells, they are destroying a lot of our natural immune responses, which is why sometimes, sadly, you'll see a lot of people who are dealing with HIV that maybe eventually leads to AIDS, that when they're getting something like pneumonia or what we call opportunistic infections, because their immune system is already so depleted, they don't have a lot of these helper T cells. That's why we see a drop in their white blood cell counts, that it's really hard for them to fight future infections as well. Oh, sorry, I just saw that question in the chat. So, yes, there is always a recording of this on, there's a YouTube channel. Let me see if I can find the link. So I found the PSU Learning Center's YouTube. And so what happens is after the stream, I download the stream and we upload it to YouTube. It'll probably not be uploaded until tomorrow. The stream on my Twitch is available, I think, for a few days. It keeps the recording. So you can totally rewatch. There's also a key, because I have a key that I'm looking at as I'm writing with you that I've done. And that should be posted after the stream, maybe the next week, under where you can find the worksheets on your Canvas page. Yeah, of course. I'm going to put the Google form in one more time. And then there is another question in the chat that asks, what results when DNA regions undergo random recombination? And is it a decrease in gene recombination? So all I meant to say was, the reason why we can produce so many receptors on B cells while not having that many genes is that there are 3 different variable regions on the antibodies, as I've shown. So those 3 parts have 3 different segments on the DNA. So what I mean by recombination is that there's a possibility for genes to switch over and recombine, and all I was trying to say by that is that it gives us even more opportunities and more kinds of AB receptors and proteins versus DNA. We only have so much DNA, but we can make a bunch of different proteins on B cells because we have recombination, we have different parts on the genes, and we have 3 different variable segments on the antibodies. So we have a lot of variability in our antibodies, which is why they're so effective to a bunch of different pathogens. Does that make sense? Okay, awesome. Okay, this was the end of this week's live stream. So I just finished off that last question, just saying writing down what I said about how it depletes our immune system. So does anybody have any questions? If not, of course, I hope you enjoy your weekend. I'll be here next week. I can't believe we're already halfway through the term again. I feel like time is flying. But yeah, thank you all. Thank you guys. Yes, of course. I love being helpful. Thank you. I really appreciate it. Yeah, of course. Thank you guys so much. You're all so sweet. Seriously. If you need any outside help too, totally feel free to email me about anything, any questions, any help with PSU. I'm all ears. I'm all here. But yeah, okay. Have a great weekend, you guys. I will see you next week. Oh, happy Earth Day! Happy Earth Day! I can't believe I forgot. Happy Earth Day. Oh, I'm very sad. I love celebrating. Anyway, okay. Bye, guys.